Yeah. So let me first introduce Dr. Werner Treptow. He got his bachelor's degree in biology in 1998. Some of the freshmen was not born at that time. We have some undergraduate students and some graduate and also postdoc and also one professor,
Okay.
including me too. And he got his PhD in molecular biology from the University of Brasilia in 2003, and then PhD in theoretical and computational chemistry in 2004. And after that, he moved to France and have postdoc training in Professor Terex's group.
Yes.
And then moved to Philadelphia and joined Dr. Michael Klein's group. So we had about 1 or 2 years overlap.
Yes.
He was a visiting professor in Dr. Klein's group. And then he got a position at the University of Brasilia as associate professor. And he has experience in computational molecular biophysics, working mainly on structure, function, and regulation of ion channels. So today he will talk about his recent work on the biological membrane. So let's welcome.
Okay, so good morning, everybody. So I'd like to start my talk by thank you, Hal, for this kind invitation. So it's a pleasure for me to be here. I'm really very happy. And it's my first time in China. I'm really impressed by this beautiful country and very nice people. And also I'd like to thank Yang for yesterday we had very great moment, right? So yeah, and today I will deliver a talk on this topic: energetics and diversity of charge transport across biological membranes. I will be talking for approximately 35-40 minutes. So we're gonna have many opportunities to understand what's going on here. So just relax, and I will give you all the details on this topic, so we can, after these 35 minutes or 40 minutes, I can give you the opportunity to ask as many questions as possible. So let's start by looking at the system. So as you know, a membrane system, so like here I show you a phospholipid bilayer, this system can store a great amount of energy in the form of this electric gradient, which is essentially associated with this reversible thermodynamic work. What is the work to insert a particle or charge in the system where the system has a specific voltage V. So essentially here I'm showing the voltage across the transmembrane system. And microscopically, in the context of molecular dynamic simulations, we can simulate a membrane system under an applied voltage by taking consideration this charge and balance protocol. So what is this? We simulate a constant volume membrane system. So here we show the membrane in presence of explicit electrolytes. So we see ions here. And under these conditions, the membrane works as a capacitor. So any charge imbalance introduced in this system may generate an applied transmembrane voltage. So if you displace ions between electrolytes, you may depolarize the membrane, as shown here. So the colors are indicating that you have a transmembrane field going across the membrane. So the voltage gradient is an energy source for a number of processes, like the insertion of charged peptides in the membrane, like the operation of voltage sensors, and the displacement of ions across ion channels. All these processes are taking place in biological membranes. So we are talking about displacement of protein charges or ion charges across the field, the transmembrane field. So necessarily we are talking about the production of gating currents or ionic currents, so currents, which can be recorded experimentally in electrophysiology. So here I show you a family of gating currents resulting from the operation of voltage sensors, for instance, or ionic currents resulting from the displacement of ions. So these are single molecule experiments. So you see you are looking at the operation of a single molecule. So eventually you can average out these currents to get average currents, so ionic and gating currents. And eventually you can, from here, get the characteristic curves. So these are very important curves. So the gating curve, gating charge versus voltage, or the conductance versus voltage curves. So these are very important quantities you can derive from the experiments. So from a theoretical point of view, we can describe the charge transport across a given reaction coordinate, let's say this transmembrane direction, in terms of this free energy profile, or the so-called potential of mean force along the reaction coordinate. So this function here is a voltage dependent function. So, and this has two major components. So the so-called chemical free energy, which is voltage independent, and this function accounts for the atom-atom interactions between the charge and the environment, plus an excess free energy that arises from the coupling of the charge Q and the applied voltage. And by coupling, I mean a specific three-dimensional function, it's a map, that reports the coupling of the local electrostatic potential with the external voltage. So what does it mean? So it means the following. If you have a transmembrane field or an electric field points down in the system, this energy stabilizes a positive charge in the interface of the membrane. Otherwise, it stabilizes outside the membrane if you have a field point out. So this construction is very important because we can link this free energy profile with a number of kinetic theories like Brownian dynamics, nucleation and growth, to derive information on these gating and ionic currents and to get these curves. And eventually we may also solve the reverse problem. We may learn details about this free energy surface by reading information from here. Okay, so that's the problem. That's the topic that we're gonna discuss today. So what we want to do in this talk today? We want to explore two aspects. So the very first one, we wanted to explore this free energy profile and its linked to this kinetic theory to study charge transport in complex systems like ion channels. And our second goal here is to explore this decomposition of the free energy surface in chemical and excess free energy to understand diversity in charge transport. So how the charge transport can become diversity in many systems. Okay, so let's go. So when you simulate a sodium channel embedded in a phospholipid bilayer, which I'm not showing here, and in presence of explicit electrolytes, so and our applied voltage, you see this spontaneous process taking place. You see ions going across the channel from one side of the membrane to the other side. So in these simulations, we may sample many crossing events. You see, here is the position of the ions going across the channel as a function of simulation time. So you can sample many crossing events. So in the context of charge and balance protocol, we may, every time an ion goes across the channel, we have to rebalance the electrolytes to keep the applied voltage constant, as shown here. So this is the transmembrane voltage in time. So fluctuations of the applied voltage store informations on the instantaneous ionic charge, and therefore on the instantaneous ionic current in time. And if you perform these simulations many times at different applied voltages, you can get this characteristic ionic current versus voltage curve, from where the slope of this curve, you get the conductance of these channels. Okay? So now, if the process, the conduction, is a Poisson process, and more importantly, if it derives from a stationary Boltzmann distribution, which means that the structure of the pore and the microstates involved in the conduction are not changing under an applied voltage, okay? Then we may rely on this construction here. We may rely on the chemical free energy profile along the reaction coordinate, plus this excess free energy for the charge along the reaction coordinate, to reconstruct this voltage-dependent free energy, as shown here. And this voltage-dependent free energy, in combination with this one-dimensional Smolchov's equation, can be used to predict current and conductance. So why? So this is the kinetic theory that connects the density, the dependence of the density of ions across the reaction coordinate with time. And as you can see here, there is an explicit dependence of the free energy profile in space. So that's why you can connect this free energy with this equation to get there. And finally, if everything is satisfied in a consistent way, we may eventually solve the inverse Boltzmann problem, where the density of ions under an applied voltage can be used to inform us the density of ions connect to this potential of mean force. Okay? So our first goal here is to see how those conditions are satisfied for a complex system like this channel. Okay. So just keep moving. I mean, we're gonna have the opportunity to see this many times. So this is the channel embedded in a phospholipid bilayer. And when you calibrate the channel by means of molecular dynamic simulations, you see that two sodium ions, can occupy the selectivity filter of the channel. What is the selectivity filter? The selectivity filter is this protein constriction here that selects sodium ions over other ionic species. So it's a very specialized structure. And essentially, the conduction process is determined by this stat here. So ions must go across this structure. So let's see what happens at 0 millivolts. So here I show you the potential of mean force, the chemical free energy profile for the two ions across the main axis of the selectivity filter, this Z direction. And we can see here that the filter provides two bind sites for sodium ions, as shown by these two minima here, right? If you expand this one-dimensional profile to the surface, now informing us the energetics for this process along this radial direction here, as shown here. So this is the main axis and this is the radius. We understand that the bind sides can be on axis, but also off axis. So we have more degrees of freedom to stabilize. And what are the colors are meaning here? The colors are describing a heat map. So yellow is the lowest free energies for the ions to bind the future. Okay? So because we have this kind of surface, then we can derive or can understand the conduction process across the future along this minimum energy pathway. So you have these two ions going across the future by adopting this single file configuration and or this side-by-side configuration until you deliver an ion across the future. So you have conduction or in that direction as well. Okay, so now let's see what happened in terms of the mechanism when you simulate the current, when you have an applied transmembrane voltage. So here what I'm showing the result of many voltage-driven simulations where we collect many crossing events across the future. And then we reconstruct the very same surfaces. So here is the probability distribution of ions in the selective future along the main axis and the radius of the pore. And if you compare this profile here, or this surface with that, derived at 0 millivolts, you see that they are pretty much similar. This is a distribution under voltages, and these are the distributions at 0 millivolts. Okay? So the consequence of this surface is that you follow the very same conduction process. There is a weak knock-on process, which means that ions can adopt these single file or side-by-side configurations. So the mechanism is pretty much the same. So what does it mean? So the weak knock-on mechanism ensures that conduction is a Poisson process, and the fact that the microstates here are pretty much the same than those observed here means that they are derived from the same stationary Boltzmann distribution. So we can apply the one-dimensional Smoluchov's equation to predict the current under an applied voltage. And here I show you preliminary results from our group. And we compare the current at 100 and 500 millivolts with the results determined or derived from Wallace and co-workers, demonstrating the very same estimates but derived from flux. So you see, these are the estimates from PMF and kinetic theory. And these compare very well with the numbers collected directly from flux simulations under voltage, meaning that the theory and the PMF construction works. And as a bonus, we can also solve the inverse problem. So here I show you, except by the difference in the bias of the voltage, I show you the superposition in space of the density derived from the potential of beam force at a given voltage and the density collected directly from the flux and the simulations, and they overlap, as shown here. So what is the major conclusion of this first part of my talk? Is that you can use this potential of beam force construction in terms of chemical and excess free energy in connection with a kinetic theory to study conduction across complex systems like this ion channel. That's the major message. So the second part of my talk, I will now explore the PMF construction to study diversity in charge transport systems. Okay? Okay? So more specifically, what we want to do. So now let's consider a general transport system involving the transport of the charge between two microscopic configurations x and y along a given reaction coordinate. The stability of this process is described by this orange potential of mean force, which derives from the free energy differences at 0 millivolts between these two states, plus an excess free energy resulting from the electrical displacement of charges at a given transmembrane voltage V. Okay, so this is the PMF decomposition that I mentioned before. So now let's consider a second system that accumulates a perturbation, star. So the perturbation star can be a mutation in the channel, can be a modification of the lipids, can be any alchemical perturbation of your system. And now we are interested to investigate this difference in free energy. So this is a different system with a different free energy landscape. So from a thermodynamical psychoanalysis, we may write this difference in terms of two components. The variations in the chemical free energy plus variations in the excess free energies.
So here you use the same reference on the other on the left side, right?
Yes, the same reference.
How can you assume the barrier is similar?
No, we are not assuming. We are just looking at the difference in the stability of the two states. So
okay.
we can still have a different barrier, yes. So we are just trying to rationalize the differences in their thermodynamic stability. So
That's
the kinetics can be very,
states.
two end states. The stability can be very different. Okay, because we can do this decomposition here, we may rationalize this difference here in terms of these two scenarios. So the first one is the so-called field reshaping. So here we are saying that this star perturbation is changing the way that the charges are coupled with the external field, but not the way that the charges are interacting with the environment. And in the second scenario, we are saying that perturbation is changing the way that the charges are interacting with the environment, but not the way that they are coupled to the external field. So we have these two opposite scenarios. So what do we want to do? We want to investigate this diversity in energetics by making a distinction between these two scenarios in terms of this contribution here. So this is the variation of the gain charge resulting from field reshaping. So essentially what we wanted to compute is B term here. And this B term here is informing us the dependence of the electrical distance profile along a given reaction coordinate with the position of the charge along the very same coordinate. So this is a complex problem. This is a self-consistent problem. Note that as the charge moves along the reaction coordinate, its coupling with the external field may also change. So that's exactly the amount of information we wanted to compute here, and to see if it's happening or not in many charge transport systems.
So the reason for the change in coupling is because of the conformational change, or?
Yes, because when the charge is displacing, so you may change the environment. So you may change the dielectric.
Okay.
So
the screening effect.
Yeah. So then you may recouple the charge in a different way. So let's see if it's happening or not in many systems appearing in biological membranes. So that's the question exactly. So let's discuss voltage sensors. So voltage sensors are 4 helical bundles They are membrane proteins, and they are very important for many biological processes, like the propagation of action potential. They can be very important in phosphatase. They can be very important proton channels. You see, in a large range of biological problems. And here I show you the operation of the voltage sensor domain of a specific potassium channel, K1.2. So when you simulate this structure under an applied voltage, you see this process, this spontaneous conformational change of the sensor. So essentially what the sensor is doing is transforming voltage differences across the membrane into molecular motions. So that's why it's a voltage sensor. So what is happening here is the following. This helix, the S4, containing 4 to 6 basic amino acids, shown here in green, which are positively charged amino acids. S4 is moving downward across the field. And by doing that, S4 is transporting some amount of charge across the transmembrane field. So S4 is generating the so-called gating charges. And in the context of this charge and balance protocol, we may compute gating charges directly from the fluctuations of the applied voltage, in the same spirit that I mentioned before. So we can define a instantaneous gating charge from where we can derive a instantaneous current. And here is a typical gating charge versus time profile you can record in this simulation. So from the structure, when S4 activates, it transports 3.2 elementary charges. This is pretty much consistent with experimental findings, measurements.
So in the meantime, the 4 negatively charged residues
remain stationary. Yes, they are not. I mean, that's not
they are not on S4.
Yes, on the only as you see here, the only moving part is
S4.
S4.
So what is the internal voltage, external voltage you applied here? What is the
So in this simulation we have a field point down. So we have an excess of positive charge outside the membrane.
The magnitude.
The magnitude. So yeah, we have simulated the channel in approximately 600 millivolts. So we start seeing the deactivation of the process in 2.2 microseconds. So you see, it's this is not physiological, but then it's to speed up the system. So the conclusions in the literature is not only my results, but we have this consensual view in the literature that the voltage sensor of K1.2 operates by this up-down cycle biased by voltage. Okay. And what about diversity? So S4 is highly conserved, but still in nature we see that voltage sensors can be very diverse. They can have different rates of activation, and they can have different voltage sensitivity, which means that the amount of gating charge they transport during the activation process can be very different. So what do we want to do? We want to take advantage of a data set of structures, of voltage sensor structures that have been published and resolved by means of different strategies like X-ray crystallography, like AMD simulations. So we wanted to export this diverse data set of structures with different sequences and different conformations to study their functional differences in terms of free energy variations. So, in here, yes?
You mentioned S4 is conserved, but how about S1 and S2? Are they quite different among different species of.
Yeah, they may accumulate mutations.
So is this the reason that the different channels have different sensitivity for the voltage?
That's the reason. I mean, you can have voltage sensors. I mean, very fast voltage sensors, slow voltage sensors, and this data set is very useful for us at this moment because they accumulate alchemical perturbations. So they have mutations in the protein environment at the vicinity of S4. So that's exactly where our theoretical framework can apply.
So is there any clear implication, I mean the correlation between the mutation or the environment of S1 and S2 versus
the speed?
Yeah.
Yes. Well, there is this beautiful paper by Pancho and Lacroix, where they take a voltage sensor of a potassium channel, which is slow, and a voltage sensor of a sodium channel, which is fast, and they identify the molecular switch to transform one voltage sensor completely. Yes. And so why they are different? So we wanted to understand if they are different because chemical free energy perturbation or an excess free energy perturbation. So the field is changing or not. So that's what we want to
know how to control the sensitivity.
Yeah,
the voltage sensitivity.
Now they know. Yeah, this is a very beautiful paper. They know. Yeah. Experimentally. Yeah. Okay. So what I'm showing here, I show here a family of electric distance profiles along this gating port axis of voltage sensors in the data set. So here I have a family of profiles, not only one, a family for those structures. And we see very clearly that there is this quite important voltage drop across the hydrophobic plug of the voltage sensor. And this drop is a consequence of these very unique hydration properties of the voltage sensor. So this is the number of water molecules hydrating the voltage sensor along the gating port axis. And the hydration structure mimics this hourglass shape here. So you have water here, you have water here, but not in the plug. So that's why you have this form for the electrical distance profile. Okay, so now, if you compute the field reshape between M2KL structures in the data set in the form of this line integral that accumulates differences in the electrical distance profile between these two structures along the gating port axis, you get this matrix. So this is field reshaping between any conformation, any isoform in the data set. And what is important here is that field reshaping is worth at most 0.15 across the data set. What does it mean? So in terms of physical units, this means that you have a change in gating charge, a maximum change in gating charge of 0.6 elementary charges. And if you convert this in terms of electrical energy at 100 millivolts, this amounts to 2.5 kT. So if you compare this number with the total amount of energy required for activation of voltage sensors, which is in the range of 15 to 20 kT, this is not that much. Meaning that there is a weak dependence of voltage coupling or electrical distance profiles with sequence and structure across the data set. Then, diversity, so that's the conclusion. So then diversity arises from modifications in the chemical free energy of the construct. So we may rationalize difference in their stability in this static field scenario. So we may summarize our findings by analyzing this continuum electrostatic model. So Okay, so now we can put all together. So this is a very important moment to pay attention. So let's see what's happened. So you see, this is a simple, a minimalist continuum electrostatic model that mimics a voltage sensor in nature. So it's a single gating particle construction that moves along a low hourglass-like dielectric featuring two bind sites. So here is the chemical free energy at 0 millivolts for this gating particle. So it's a bistable system. So when you depolarize, you then may stabilize. When you depolarize the membrane, you may then stabilize the particle in the down state or in the up state. And if you combine these two contributions, you get this resulting free energy landscape, which is voltage dependent. And from where you can get the thermodynamics for this particle displacement, like the characteristic gating charge versus voltage. You have this S-shaped function. So which means that in average, when you hyperpolarize the membrane, the particles stay here, and there is no gating charge. And when you depolarize the membrane, in average, the particle is here, and then you have gating charge. Okay, so now let's generate diversity. And our results are giving us a root for this. So let's generate diversity by doing an alchemical perturbation in the environment of the gating charge. So let's say, let's neutralize this bind site. Now, this perturbation changed the chemical free energy profile. It destabilized this up state, but it's not changing the shape of the low dielectric. So it's not changing the excess free energy profiles. But the overall effect is what? Is a different combined free energy landscape. And the result here is a different construct that now has this QV relationship shift rightward. So this is a voltage sensor with a smaller voltage sensitivity than the Y type. So that's the major conclusion we got for voltage sensors. So now I would like to show you one more example. So what time we have? So we still have time?
No, you still have enough.
Okay. So let's keep moving. So now let's look at a completely different system. So the pore domain of a potassium channel. So this is a specific channel, the meth-K channel. So when you simulate the meth-K channel embedded in a phosphorylated bilayer, and under an applied transmembrane voltage, you see this spontaneous process taking place. You see potassium ions, you see, going across the selective filter. And you have the transport of ions. So, again, from the fluctuations of the applied transmembrane voltage, we get information on the current and conductance.
But here you always assume a linear relationship between the current and the current and the voltage, right? Or
not necessarily, but for this is experimental, and normally for ohmic system this is linear.
almost.
Yeah,
Which means the conformational change is very small.
Very small when the channel is open. Yeah.
Okay.
So the rate limit step for this conduction process is again the transport of ions across the selective filter. So here I'm only showing two subunits of this highly specialized structure. So you see I'm just zooming here and looking what's happening during the simulation. So this filter is very specialized because it selects potassium over other ionic species. And doesn't matter how long you follow this process, you always end up with this up-down transition. So here is a knock-on process where an external ion hits the ions in the selective filter and triggers conduction. Okay? So alternative mechanisms involving side-by-side ion configurations cannot be excluded at high voltages. But this mechanism appears to be a very important mechanism at 0 millivolts or at least near equilibrium conditions. So let's take in consideration this process to understand diversity in the conduction across potassium channels. Okay, and what about diversity? So again, the very same problem. So conduction can be very different in these potassium chain structures, meaning that we have functional differences and therefore differences in their free energy landscapes. And so what do you want to do? You want to take advantage of these crystallized structures to investigate diversity. So here I show you the electrical distance profile along the main permeation pathway of two distinct potassium channels, case SA and case 1.2. So the electrical distance profiles can be very different from voltage sensor, but still we identify quite important voltage drop across the selective filter. And why? Again, resulting from this very unique hydration structure. So the filter is dehydrated. So if you compute field reshaping between these two channels, and you convert field reshaping in terms of excess free energy differences, we conclude the following. Field reshaping is minimal. So there is a weak dependence of voltage coupling, electrical distance profiles with K-chain structures. So diversity in their conductances must derive from changes in the chemical free energies.
So here you calculate the profile for the dielectric constant. Depends on the crystal structure or depends on the simulation trajectory?
That's an average profile derived from the conformation, the equilibrium conformation and the simulations. Yes, but which is essentially the very same conformation of the X-ray structures.
Okay. And how did you estimate the error for, I mean...
The fluctuations, yeah. Right? Well, I'm not showing here, but this is very conservative profile. Okay. Yeah, the structure is fluctuated, but always you have these water molecules, at least in the open conductive conformation, which is the case here. Always you have this water and that dehydration, so the shape of the dielectric is pretty much conserved in simulation time. Yeah. Okay, so now let's see the energetics, the chemical free energy profiles for the up-down transition across the selective filter of case SA, shown here in black, and case 1.2, shown here in red. So here specifically we use these Jarzynski's identity and distribution of forward and backward irreversible works to derive this potential of mean force. So this is the chemical potential of mean force at 0 millivolts, right? And you see clear differences between the two channels. So for case SA, up and down configurations have about the same stability, but not for case 1.2. And what is happening?
So when you apply Jarzynski identity to explore the free energy profiles, you need to hundreds of thousands of sampling on the same system to get the ensemble average, right?
Yes, if you're far from equilibrium, probably yes. If you're near equilibrium, as our case here, we have this convergent PMF with, like you see, a not that large distribution of.
So how many cycles did you use to do the?
This is a paper that he published in 2006. At the moment here, I think we have like 20 forward and 20 backward transitions. But at the time, the speed for the transformations, we learned from equilibrium transitions. See, so we were not that far away from the.
So as long as the structure is near the equilibrated structure, it's fine.
It's fine. No, the structure is completely fine. We see this forward and backward transition. So as shown here, see you have the coupling of the. This is actually the trajectory in time of water, potassium, water. And this is the spread of the distributions from where we got the work. Okay, so what is happening here? You see, the reason for that change is the mutations. So this is the structure of cave 1.2, and when compared to case SA, this structure accumulates one or more mutations in the environment of the charge transport. And we can convert the potential of mean force of cave 1.2, shown here in red, the forward PMF, shown here in red,